So, all right, we are recording. Where we left off yesterday is classification of neurons, and we covered the structural classifications, the three groups, which is multipolar, bipolar, unipolar, and that is just based on basically, obviously, structural characteristics of that neuron. And what I always tell students is that's what it looks like. Easy way to think about it. I'm going to quickly review functional classification of neurons. The reason I say quickly is because not only do we have a lot to talk about today, but you already know basically what this is. So I'm going to call up the whiteboard and just write down functional classifications. That's functional classification of neurons. And you've already heard this language and you know what they are, but I'm going to quickly go over them again. First one, sensory afferent. These carry impulses toward the central nervous system. That's first and foremost. And so I'm just going to write this, toward CNS. These tend to be unipolar. Now we know what that means. It means they tend to have one process attached to the cell body. The exception to that is special senses. So I'm going to put plus special senses, which are bipolar. But those are rare. Those are not common in the human body. Also with the sensory afferent neurons, their bodies, their cell bodies are found in clusters called ganglia. I mentioned that term yesterday. In the peripheral nervous system, okay? So let's go to number 2. We know this one as motor efferent. If you hear me say efferent, that's just because that's the way, when I was a student, I was taught to say it. But I believe the correct pronunciation is efferent. These carry impulses away from the CNS. That's first and foremost. And they carry impulses to what we call effectors, which would be muscles and glands, effector organs. These tend to be multipolar in structural characteristic or structural classification. And their cell bodies tend to be found in clusters called nuclei. That's N-U-C-L-E-I, inside the central nervous system. So even though motor neurons, if you're confused a little bit, how are their bodies found in the central nervous system? If a motor neuron goes all the way out to your toes, for example, to regulate a muscle contraction, the cell bodies of these multipolar neurons are way in the central nervous system, either the brain or the spinal cord, and then their long axons are what extend way out into the peripheral nervous system. So those motor neurons tend to have, depending on where they need to, what they need to innervate, what their effector organ is, they can be very, very long. All right, number 3, functional classification are called interneurons. These are also called association neurons. So if you see that term, same thing. And And these act as, because we're talking about functional classification, they act as a bridge between number 1 and number 2. These tend to be the 99% of all functional neurons in the human body. These are found in the central nervous system. So these interneurons, let me just classify these. Interneurons tend to be multipolar. Motor neurons also tend to be multipolar in structural classification. And then sensories tend to be uni and bipolar. All right, I'm going to move on from this. If you have any questions, just ask, but I really need to get into this. Most of this next bit of what I'm going to talk about is wildly complicated, and I'm going to try to simplify it for you. It is sort of the nuts and bolts of how the nervous system works, and how a neuron, an individual neuron, works. And it's very complicated. The language is difficult if it's the first time you've heard this. If it isn't, then you'll have an easier time. We need to go into the next section of the chapter and first talk about membrane potential. It's a lot of crazy detail. I'm going to simplify it for you so you understand it. My advice is watch this lecture multiple times to get a bit of a Reader's Digest version, a simplified version. Then you'll understand the overall story, the overall picture. You can layer on detail after that. So what this topic refers to, membrane potential, is the cellular plasma membrane, in this case of a neuron. And there are 3 types of gated channels along the membrane of a cell. We're mostly interested in 2 of those 3, but I'm just going to mention all 3 types. The first one is chemically gated. The second one is voltage gated. It's supposed to be an O. And the third one is mechanically gated. I'll just explain what those mean. In this instance, we are mostly concerned with number 1 and number 2. All this means is there are little gates on the plasma membrane of a cell. There are protein channels that allow things to move in and allow things to move out of a cell. But there are different types of protein gates or channels. Chemically gated channels are open and closed when a chemical binds to either a protein on that membrane or the gate itself. Voltage gated channels just means those open and close, these gates, these protein gates open and close when there's a change in electrical activity of this membrane. And mechanically gated channels open and close to allow things to pass through when physical depression is put on it, when there's some sort of physical contact. So these we're not going to talk a lot about right now. They tend to be things like touch receptors in the human body. And when you touch something, there's a change in opening and closing of things across this membrane. We are going to talk about chemically gated and voltage gated, particularly voltage gated right off the bat. So just to build up a vocabulary of how a neuron is actually going to fire an electrical impulse, we need to define some things. So I'm going to write resting membrane potential. We're building up sort of a vocabulary list, a dictionary of how we're going to understand this material. Technically, this resting membrane potential, what that means is when a cell is at rest, it's not doing anything, it refers to the difference in ionic potential across the membrane of the cell. So, if this is the membrane of a cell and it's at rest, the first thing we're going to establish is there's a difference in the environment outside of the cell from inside of the cell. Okay? What creates that difference is ion concentration is different out here than it is in here. And the difference in the ion concentration is going to create a voltage difference, a charge difference. So the ion distribution in resting membrane potential of a nerve cell is this. The cell is high in sodium ions outside of the cell, and it's high in potassium ions inside the cell. So at rest, a nerve cell, and by the way, most cells in the human body have this same ionic distribution. There are a lot of sodium ions, and we're just talking about the environment right along the edge of this cell. It doesn't mean that way out here, there's also a lot of sodium ions, and way deep in here, there's lots of potassium ions. It just means right along this edge, there's lots of sodium ions outside, lots of potassium ions inside. What that does is it creates a difference in charge environment outside and inside, okay? So sodium is more positive than potassium. They both have plus charges next to them. Sorry, there was a neighbor that I had to close the window. Sodium has a positive charge. Potassium also has a positive charge. But sodium has a much greater positivity. It's stronger. Its positivity is stronger than potassium. And so we have a high concentration of sodium out here, a higher concentration of potassium in here. That creates a net charge of a positive environment out here, and relative to the inside, it's negative inside. Don't get so hung up on that this is positive, how can it be positive out here and negative in here? Really, what it means is the outside of the cell is more positive than the inside. And the reason is because sodium has a higher strength and positivity, that's all. So I'm making a mess here. I'm going to go to another board and just continue and clean up my diagram. So at resting membrane potential, just remember this. There's a high concentration of sodium ions out there's a high concentration of potassium ions in. This creates a net positive charge outside of the cell and a net charge of negativity inside of the cell. That's just a relationship. Whenever we have a side of an object, or a cell in this case, that's different in charge compared to the other side, that creates the potential to do work. So when we talk about membrane potential, the cell either has potential for energy or to do work, or it has very little. The more we separate a polar environment, the more potential there is for something to happen, for work to be done. It's just like if you have a tennis ball resting on the floor. There's very low potential for it to do work. But if you lift it up, the higher you lift it off the floor, there's more potential for something to happen, because if you drop that tennis ball, the higher you raise it, the more that gravity is going to want to pull it back to the floor. That's a crude example that sometimes books use. Just remember these keys: sodium out, potassium in, positive out, negative in. With that positive negative relationship, we call this a polar environment. Polar just means opposite. So at resting membrane potential, this, positive, negative. Now we can continue our conversation. We say that if we have opposite environments on one side from the other, it is polar. If we depolarize that environment, I'm going to write depolarization. Depolarization means there is a decrease in membrane potential. That's what the word depolarization actually means. So the inside of the cell becomes less negative. Now, we know what these words sort of mean. We know what polar means. We know what depolarizing. Let me draw a picture. If we have positive out, negative in, if this environment of this opposite-sided membrane, if positivity leaks this way and negativity leaks this way, we are depolarizing that membrane. Okay? The whole reason that a battery that you put into your phone or that you use to energize something, the whole reason it has the ability to do work is because it has a positive end and a negative end. You know that. When you hook something up to this battery, you are going through the act of depolarizing these opposite ends. It brings these opposite sides, the ions in this battery, closer together. That's an act of depolarizing. That's what a membrane does on a nerve cell. Now, I need to define this term, a graded potential. Don't get hung up when you're reading the chapter in your book on all of the electrical speak in there, meaning millivolts, volts, charges, current, all of that. That is, in my opinion, That's fluff with regards to how a nerve cell really works. So if you don't understand all of the speak about volts, millivolts, charge, current, electrical current, this is not a class in electricity. It's not a physics class. This is a class in human anatomy and physiology. So I'm more concerned that you understand how a nerve cell works, not how electricity works. Having said that, what the definition of a graded potential is, we're still building vocabulary here, a localized, short-lived change in membrane potential. A localized, short-lived change in membrane potential. We say that it's graded, this word, the first word graded, because the magnitude of the depolarization of the change in membrane potential is directly related to stimulus strength. So I'm just going to write the word graded in quotes. It's graded because the magnitude is directly related to stimulus strength. Now we're going to use some of these words. Graded potentials occur at or around the soma, the body of the nerve cell, including dendrites. So they occur at or around the soma of the cell. If a graded potential, let me go to another board here. I'm going to draw a picture and explain this to you, hopefully, in picture form after we write some words. So if a graded potential reaches the receiving cell's threshold, I'll define this later, then the neuron will generate its own action potential. Whenever you see action potential, think electrical stimulus. Now this action potential occurs, this starts at the axon hillock of the neuron. It's called the trigger area of the axon hillock, but we learned where this is on our anatomy of a neuron, so I'm just going to use the term axon hillock. Also, an action potential is considered an all or none response. That means once it starts, it can't be stopped. It fires all the way. That's what all or none response means. It either happens completely or not at all. Now, the rest of this lecture is probably going to be sorting all this business out. If you're confused, that's okay. You probably should be at this point. I'm going to try to unconfuse you now with the rest of this bit. So your book goes through this in step fashion, which is good. I'm going to try to do the same, and I'm just going to title this, these are steps to the generation and propagation of an action potential. Now we're going to decipher some of these words that we just learned, some of these terms, and their actions and how they affect a nerve cell. So first thing I'm going to do is draw a typical motor neuron. I'm not going to myelinate the axon, because that would just mess up my drawing right now. So down here at the soma end, cell body, we have lots of dendrites. We have, I'm just going to put a nucleus in here so that you remember this is the soma, the cell body. I'm not going to draw any other organelles. All right, so this is the receiving end of the cell down here. When this nerve cell is just sitting there, it's actually not, when it's at rest, it's not doing nothing. What it's doing is it's manufacturing neurotransmitter, chemical. And that manufacturing process is sending raw material for neurotransmitter to be assembled and stored in these axon terminals, in those little bubbles called vesicles. So our nerve cells, even though they're not firing an electrical impulse all the time, they are active. And what they're doing is they're creating and storing neurotransmitter down here. But when this nerve cell does fire an electrical impulse, here's the sequence of events. First, at resting membrane potential, we know that sodium concentration is high outside of the cell and potassium concentration is high inside of the cell. We've already established that. I'm not going to draw it, but that goes all the way around the cell, including down the axon, this separation of ions. We've also established this, that the net charge distribution created by this separation of ions is positive out and negative in. And that's also true all the way around the cell. So when this cell is at rest, we have high sodium outside, high potassium inside, positive out, net charge, negative net charge inside the cell. There's a protein pump on the membrane, all along the membrane of most of our cells, not just nerve cells, that creates this. It pumps sodium ions out here and potassium ions in here. And it uses ATP, it uses a lot of energy, and it creates this polarized gradient. Now, step two. So step one is just called resting membrane potential. We've established that. And it's the sodium-potassium pump that sets up that gradient and that charge distribution. It's a polar environment. Step two, depolarization. This is a complicated step. I'm going to explain this now on my picture. If this nerve cell that's currently at rest gets stimulated, let's say by another nerve cell, so I'm going to put an axon coming from this direction. There's its axon terminals. If this nerve cell off to the bottom releases neurotransmitter, and that neurotransmitter binds to receptors, I'll use red, since I used black for the that sodium-potassium pump. Let's use red. So these receptors all along the soma and dendrites can receive neurotransmitter. If these receptors receive neurotransmitter, it causes a change. Let me go back to black. It causes a change in the polarization along here. We call this depolarization because what happens is sodium gates, when this neurotransmitter binds to these receptors, sodium gates, those protein gates, open up and sodium leaks into the cell. That's an act of depolarizing the cell. Remember, polar opposites means positive, negative. If sodium ions leak into the cell, we are causing this ionic difference to get closer to zero, not further apart, but closer together. So, and this act of depolarizing the cell is first a graded potential around the cell body, or soma. So there, when this cell, the nerve cell that I've drawn, is stimulated, it receives neurotransmitter, we get ripples of depolarization around the cell body. That means sodium leaks in a little bit all around this cell body. And it's a graded potential. That means depending on this stimulus strength that it's getting, that it's receiving, these waves of depolarizing will go further and further across the soma. So if this gets a mild stimulation, there's just little ripples that die out, little ripples of depolarization that die out. If we give this a greater stimulus, this resting neuron, there are bigger waves of graded potential depolarization that extend further. Think of it this way: when you throw a pebble into a resting pond, if it's a small pebble, you get small ripples that die out quickly. If you throw a larger rock into that pond, you get larger waves that go further. Same here. If you stimulate this gently, you get small waves of depolarization that die out quickly. If you stimulate this really hard, you get larger waves of depolarization that extend further. These are graded potentials because they are dependent on stimulus strength. Now, if we stimulate this nerve cell high enough, strongly enough, the graded potentials will cause this receiving cell to reach its threshold. That means these waves of depolarization will be so strong that they will cause something to happen here, something magical to happen here at the axon hillock. Okay? So depolarization just means we stimulate this resting cell. If we stimulate and it causes it to exhibit a graded potential, if we stimulate this resting cell strongly enough, we get enough depolarization through graded potentials that it will reach this resting cell's threshold. And we've defined threshold as the point where this cell will fire its own action potential. And once that happens, we can't stop it. It's an all or none response. So let me clean this up a little bit. I'm not going to delete this. I'm just going to add on top. So where are we? I just kind of made a mess with all my drawing. Here's our resting neuron. We have at resting membrane potential, we have sodium ions out, potassium ions in. We've got a positive environment outside, a negative environment in. When we stimulate this cell, if we stimulate this cell gently around the cell body area, we get waves, small waves of depolarization that die out quickly. What is depolarization? It's the flipping of this charge. If we stimulate this cell super strongly, we get larger waves of depolarization, the flipping of that charge, that radiate more strongly and further around the cell body. When they reach this resting cell's threshold, at the axon hillock here, sodium gates, channels, open up very widely. It's like cracking open a dam that's dammed up a bunch of water. And what happens is sodium ions gush into the cell. This is an act of generating an action potential. So we have mild waves of depolarization. If those reach a stimulus strength that's strongly enough, these sodium channels open wide. Sodium ions rush into the cell. Once that happens, we turn on an event that can't be stopped. And that event goes like this. After these two sodium gates open up, they cause the next set of sodium gates to open, and then the next ones to open all the way down this axon. Okay? In a linear fashion. It's just the same as if we have a bunch of dominoes stacked up in a row. They have the potential to fall over. If we tip that first domino over, it causes an event. They all tip over, and we can't really stop it. If you've ever set up a whole row of dominoes and tipped the first one over, it moves so fast in one direction, you can't really stop the event. Same thing happening here. Once we tip this first domino, all of these sodium gates open one after the other in a linear fashion, and sodium ions gush into the cell in linear fashion. And by the way, this is the action potential firing. Electrically, what we're doing is we're flipping the outside positivity, inside negativity. We're flipping that backwards. By all of this positive ions flowing in, we create this as a positive atmosphere, and this becomes negative. When we flip that charge, that causes this electrical event to occur all the way down the membrane. When it gets to the end, it causes neurotransmitter to be released from these vesicles. All right, so the act of the cell actually depolarizing means these sodium gates open, sodium ions rush in. Remember, the original resting charge was positive out, negative in. When sodium gushes into the cell, it flips the charge to be positive in, negative out. That in itself is what depolarizing means. It means flipping the charge. That also is what the action potential is, an electrical impulse of flipping this charge from positive out, negative in, to positive in, negative out. And it occurs by sodium ions rushing into the cell, gate after gate after gate, all the way down. So that is this cascading of sodium ions rushing in all the way down the axon is the firing of an action potential. Now, step 3 of this process is called repolarization. Not quite finished with the story. We know back here at the beginning of the story we had sodium out, potassium in, positive out, negative in. If we give a strong enough graded potential that it depolarizes this membrane enough to reach this cell's threshold, at the axon terminal, large sodium channels open, sodium rushes in. We cause an action potential all the way down the cell that we can't stop. Now, as these gates open one by one, the sodium channels all the way down the axon, these gates upstream start to close. They close shut after they've opened. So as these doors are opening, the doors behind them start to close, and then potassium gates open. So after these sodium channels have opened and sodium has rushed in, they close up, and then potassium gates, I'll label those yellow. Potassium gates along this same membrane open up, and potassium rushes out of the cell. This is called repolarization. So during repolarization, first thing you need to think of is potassium ions rush out of the cell, and enough of them rush out of the cell. Remember, they're positive as well. Enough of them rush out of the cell to repolarize the membrane, to cause a positive environment out here again and a negative environment in here again. So I'm just going to say enough rush out to, that's why we call it repolarization, to repolarize the membrane. That means establish positive out and negative in. So during the act of repolarizing the cell, and this happens after depolarization, potassium rushes out. Enough of that potassium rushes out of the cell that we get positive outside, negative in again. So we've repolarized the membrane. The problem is we're still not back to the beginning of the story, because now we've got all these potassium ions outside, sodium ions inside, even though we've repolarized the membrane. I'm going to say, but we still need to reestablish the original ion concentration. We've established the original electrical polar differences, but now we've got flipped. We've got potassium out here, sodium inside. Before I go on to reestablishing the ion concentration, this word, just so you know what it means, and I might ask you a question on the quiz about this, hyperpolarization, oftentimes in the human body occurs during the act of repolarization. Sometimes, I should say oftentimes, because it's not uncommon. I'm going to say it's hard to put the brakes on this repolarization. It's hard to put the brakes on potassium rushing out of the cell. Sometimes too much potassium rushes out during repolarization. And when that happens, we cause even a stronger positive atmosphere out here compared to in here than there was at the beginning of the story. The membrane does eventually right itself. So let's just say we've got a certain difference at the beginning of the story of positive out, negative in. When during repolarization, when these potassium ions are rushing out, it just means that sometimes the original numbers, levels get overshot because so many potassium ions rush out. So I'm going to put causing a more positive environment outside than we had at the beginning of the story. So I just wanted you to recognize this word. It really means the overshooting of repolarization. Okay, so now we've established positive out again, and we have negative in, but now we have these ions, potassium out and sodium ions in here. It's the sodium-potassium pump that I mentioned at the beginning of the story. That reestablishes the original ion concentration. Sorry, spell that right. Of sodium outside the cell and potassium inside. So after repolarization happens, the sodium-potassium pump eventually reestablishes the original ion concentration. Now, before we totally leave this topic, I want to define refractory periods. There's two different types of refractory period. Something called absolute refractory period. And this is defined as during the action potential, that means during depolarization, during this event, when the sodium gates are open and sodium is rushing into the cell, the neuron will not respond to another stimulus. No matter how strong it is. So the reason is because the cell's busy. It's busy firing its own action potential. So during that time, during depolarization, what this does is it helps ensure one-way transmission of that action potential. So this cell, this nerve cell, when it's depolarizing, firing an action potential down its axon, if we stimulate that cell again, it won't respond to that stimulus when these sodium gates are opening and sodium is rushing in. But there is something called relative refractory period. And this is defined as during repolarization, the neuron's threshold for the stimulus is higher, but it re-exists. So I'm going to leave off the neuron's threshold for the stimulus is higher and just say, if we stimulate the cell, the neuron, during repolarization with a higher stimulus than the first one, it will respond. So during depolarization during an action potential firing, no matter what we do to the cell as far as stimulating it, nothing will happen. It won't fire another action potential. However, during repolarization, when those potassium gates, sodium gates are closed now, and when potassium gates start to open, if we stimulate the cell again with a stronger stimulus than the first one, it will fire another action potential during repolarization. So that's the difference between absolute refractory period and relative refractory period. All right, let's get to the end of this lecture. And I just need to talk about conduction velocity. The rate of impulse of propagation, that means how fast a nerve fires its electrical impulse. How fast the action potential travels along that axon. That conduction velocity is dependent on two things. First, the size or diameter of the axon. Not necessarily how long it is, but how thick it is. The thicker in diameter the axon is, the faster it will fire an action potential. And obviously the opposite is true. The thinner in diameter is, the slower it is. So first, what dictates speed? The diameter of the axon. And the second characteristic is the degree of myelination. I'm just going to write more myelin. That insulation that wraps around the axon, more myelin also equals a faster action potential firing. So let's just now classify different groups of nerve fibers based on speed. This is based on speed of transmission. And we just classify these as group A, group B, and group C. So group A fibers these are what we call somatic, sensory, and motor fibers. It's an and sign. These are the largest in diameter and they're heavily myelinated. So obviously they are the fastest. They can conduct speeds up to about 300 miles per hour. That's really fast. If you think about transmitting a neural impulse, say from your spinal cord even out to your hand, and it's moving at 300 miles per hour, it gets there really quickly. That's group A fibers. I'm going to go to a new page and just write group B. These tend to be smaller in diameter than group A, and also lightly myelinated. And they conduct speeds up to about 30 miles per hour. The easy way to remember the speeds are they're about 10-fold from each other. So group C fibers would be the smallest in diameter. They have no myelination. No myelin wrapped around their axons, and they conduct speeds up to about 2 to 3 miles per hour. Now, I did say about group A fibers that they are somatic sensory and motor fibers. Group B and C fibers tend to be associated with the autonomic nervous system. So I'm just going to say these tend to be autonomic visceral fibers. And they tend to be slower. I think of autonomic visceral as like the nerve impulses that are controlling, say, your digestive system, or nerve impulses that might be controlling like blood pressure, internal functioning of the body. Those tend to happen on a much slower rate than somatic fibers, sensory and somatic fibers. When you know this, when you touch something scalding hot, it doesn't take you very long to jerk your hand back from that. Those are the fastest, heavily myelinated fibers in the body for survival purposes. Last bit, and then I'm done. I want you to be familiar with this term, continuous conduction. This is how unmyelinated fibers must conduct their action potentials. I'll explain after I write something. So unmyelinated fibers conduct their impulses, their action potentials with continuous conduction. And that means they go, because they're unmyelinated, that means their action potentials have to go like dominoes, gate to gate to gate to gate, one after the other. It's like walking 10 feet from one point to another on the floor, placing your feet heel toe, heel toe, heel toe. All right? Now, that's opposed to something called saltatory conduction. This is how myelinated nerve fibers conduct their impulses. And how they conduct their impulses is the depolarization, the action potential, literally jumps from node of Ranvier to node of Ranvier. Remember those gaps in the myelin sheath? So I'll show you how saltatory conduction works. Continuous, I already described, it has to go gate to gate, heel toe, heel toe. If we myelinate this nerve fiber, we wrap that lipid-based insulation around it like this, and those gaps in the myelin sheath right here are called nodes of Ranvier. The reason a myelinated fiber can conduct impulses so much faster is when that action potential first fires here at the axon hillock, that depolarization, that action potential can literally jump from node to node. So rather than going heel toe from point A to point B, it literally takes giant steps really quickly. and it can move a lot faster. So that's how myelination increases the speed of transmission. All right, guys, I'm done for the day. I know this was crazy, confusing stuff, and complicated as well. I highly recommend that you let's see here. I highly recommend, I'm just going to try to get my head back so that you can see me when I talk. I highly recommend that you read your book, but also when you read your book, focus on what I talked about if you can. Watch this video again if you can. I will post it today. Don't get hung up on the details that I didn't mention in my lecture, okay? What's most important to me that you learn is the basics of how this system works, how this action potential really works. Then go to your book. After you watch this video once or twice, then go to your book. Forget about things like millivolts, resistance, charge so much. That's not the meat of it. That's just fluff. What is important is how the separation of charge, resting membrane potential, then the active depolarization, the active repolarization, and then getting back to not only resting membrane potential, but the original ion distribution charge. So I'll try to do a quick review tomorrow of this talk. It's going to go very fast. You guys did really well for showing up today. I'll do a quick 5-minute review tomorrow. You can ask me any questions you want if you have any questions about this process, and I'm happy to answer them. So I'm going to stop my recording right now.